So in this video, what we're going to talk about is protein synthesis. Now, one of the most important things we need to talk about first in protein synthesis is a molecule known as RNA. Now, RNA plays an incredibly crucial role in how protein synthesis works. Now, to understand how it does that role, we need to talk a little bit about what RNA is. So, RNA is very similar to DNA, but it has some very specific structural differences. First of all, it's only one strand. DNA is double-stranded, which means you have pairs lined up with each other, across from each other, and it's all kind of twisted up in two strands. RNA is only one strand. Part of this structural difference is because it contains the sugar ribose instead of deoxyribose, which is actually how you get the name of RNA, ribonucleic acid. One of the bases, the nitrogenous bases, is actually different in RNA compared to DNA. RNA contains uracil, which is a slightly different shaped molecule compared to thymine, which we find in DNA. Now, RNA is mostly found in the cytoplasm, which is a little bit different, where DNA is basically strictly found inside the nucleus. RNA we find a little bit in the nucleus and a little bit in the cytoplasm. So it's kind of all over the place, which again allows it to do its job. Okay. And lastly, the big thing is what its job actually is. DNA simply stores information. RNA is going to do a lot of the work. RNA is going to be there to transport that information from the DNA to where the proteins are being made. Now, there's three types of RNA that we're ever going to talk about, and really one that we're really going to talk about. mRNA is messenger RNA. That's the one that you're going to hear about almost exclusively when we talk about it in class. The mRNA takes information from DNA in the nucleus and it brings it to the ribosome where protein synthesis is going to happen. The other types are tRNA and rRNA. tRNA stands for transfer RNA. Now, this is the one we'll kind of talk about a little bit. The tRNA molecules are actually responsible for moving the amino acids into their correct location during protein synthesis. They're kind of like a little taxi service. So tRNA stands for transfer RNA, which is what they're doing. They're transferring the amino acids into their correct location. Lastly is rRNA, and rRNA is ribosomal RNA. And basically all that is the stuff that makes up the ribosome. Okay, so it's kind of weird thinking of an organelle being made up of, one component, the RNA, but that's kind of what the ribosome is. Now we know a little bit about RNA, we can actually start talking about what protein synthesis is. And you can kind of break it up into a few steps. I've broken it up into 3 for us. You can break it up into more. You can probably lump them into 2 big steps, but there's 3 right here that we can look at. The first thing that happens is that we need to unzip the DNA. Okay, so the DNA molecule, when it's closed, looks kind of like a ladder or a zipper. To access the information, we have to separate the base pairs so we can actually read the information. It's kind of like opening a book in order to read it. Okay, what happens here is an enzyme known as helicase. Okay, kind of makes sense because DNA is a helix. That's its shape. So helicase causes the DNA molecule to separate or unzip. Okay, what this does is it leaves 2 sides of the nitrogenous bases, those 2 sides of the pairs, exposed, so information can be read from that DNA molecule. So it looks basically like this. It's not a perfect representation, obviously, but that protein that it says there is the helicase, and it comes in and it unwinds and separates the DNA molecule. And you can see on each side there are some exposed bases. Okay, so that's where we have to start with, is that helicase enzyme is going to separate the DNA so it can be read. The next process is known as transcription. Okay, now transcription, guys, is very important. You're going to have to know this term. And if you break down what the term means, it kind of makes sense. To transcribe something is to write it over again. And that's what transcription is doing. It's kind of writing the information in the language of mRNA. So what will happen is the mRNA nucleotides are going to come in and bind to the sections of DNA that are there. Once this happens, it's basically made a copy of that information. Now, the mRNA molecules are going to be read in 3 base units, and we call those codons. So maybe you have a C, a G, and an A. Okay, that would be one codon, and we read that, and that's how we find the information. Once the whole molecule is made of mRNA, it's copied whatever gene it's supposed to copy, it's going to take that information, separate from the DNA, and actually leave the nucleus on its way to the ribosome. So again, visually, this is kind of what it looks like. We have an enzyme in there called RNA polymerase, and that's going to help speed up the process of creating the mRNA molecule. But we'll notice that we have the DNA separated. The RNA is copying one side of that DNA strand, and it's copying it by putting the opposite letters or the opposite nitrogenous bases with the exposed DNA and making a strand of RNA from that. Right? The one thing you will notice now that is the RNA now has U's instead of T's. Remember we said that it has a uracil instead of thymine. So now the A in the DNA bonds with a U in RNA, and everything else bonds the same way. So that's transcription. The last process is known as translation. And what happens here is when the mRNA reaches the ribosome, it binds with it, and it basically starts reading it. So what happens is the tRNA molecules match up with the codons on the mRNA molecules, and they bring the specific amino acid that they're supposed to bring. And the amino acids are going to assemble themselves at the ribosome in a specific order. The order of those amino acids is going to determine the shape of the protein, and the shape of the protein determines what that protein is actually going to do, its function. Okay? Once the protein is formed, the protein exits, the RNA molecules break up into nucleotides and wait to be used again. So again, we have a little picture here. We can see the tRNA molecule 1 coming in with an amino acid there that's ready to be kind of hooked up. One that has just hooked up its amino acid that's leaving, and a couple more that are currently bonded to the RNA that are connecting those amino acids together. Now, the amino acids, we can have chains that are hundreds, if not thousands, of amino acids long to make a protein. Most of the stuff we're going to see in our examples are pretty short because, frankly, we're not going to take the time to do all that. But this is how protein synthesis works. You start with the information in the DNA, you copy that into RNA, and the RNA uses that as a template to put the amino acids in their correct sequence.